Hi Dan, are you there?
Hello, I am here indeed.
Okay,
Sorry, I was
great.
dealing with a mini toddler crisis.
Okay. And if you
Um,
can switch on your camera also, that would be great.
yes, let me just
Works.
sort out a couple of things.
Perfect.
Okay, well, hopefully you can all see me. I'm not sure what else you can see.
Yeah.
Okay, so shall I take it away?
Yes, please.
Okay,
That's
well, firstly, hello everyone, welcome. It's really nice to be here and to be with you for this week. My name's Dan Depledge. I'm Assistant Professor in the Department of Medicine here at NYU. I'd probably describe myself as a systems virologist. I work in the field of virology, and mostly I try to tackle interesting virological questions from a point of view of computational biology, and also through analysis of high throughput sequencing data sets, and then some good old-fashioned lab biology. The purpose of the next stage of this course really is to introduce you to the idea of sequencing informatics. We're going to be working on this over weeks together. The course structure is going to look something like this. I'm going to get my screen to advance. This week I will be introducing you to the major sequencing technologies that are out there, a little bit about how they work and the sorts of applications they're useful for. And then later in the week, I'll be talking to you a bit about file formats. This sounds incredibly dull, but it's actually a hugely essential part of understanding what high throughput sequencing data looks like, and how you can work with it, and how you can tell the difference between Illumina data and Nanopore data, PacBio data, and so on. Then next week, we've got Angelina Volkova taking over. She's going to be giving you tutorials on how to use Linux, which is absolutely crucial if you want to work on sequencing informatics. Then after that, we have Elizabeth Katz-Nelson, who's going to talk about processing quality control, how to download data, where to find data and download it, and how to do trimming of data. This will make more sense as you advance through the course. Then Anna Yeaton will take over and be talking about how do you actually align sequence data against a genome of interest or a transcriptome of interest, and how do you visualize this in ways that are suitable for publications, but also just in terms of satisfying yourself that the data have aligned correctly and allowing you to look at regions of interest within the genome. Then Igor Dolgolev will take over and give you a primer on RNA sequencing. Obviously, RNA sequencing is a popular topic, but it's also a very big topic. So we won't be able to cover everything, but hopefully it will be enough to get you up and running in the kind of the basics of how to take data and generate generate counts of gene expression and how to perform differential analysis of these. And then finally, Macintosh Cornwall will be going deeper into gene expression analysis. And I think we'll be using programs like DSeq and EdgeR, which are the popular implementations. Before we get started, I also need to plug a couple of courses I'm involved in that are available through the Biomedical Informatics program. One of these is Applied Sequencing Informatics, which at the moment is running from January to May each year. It is mostly aimed at the people coming on the master's program, but we have a lot of grad students and some postdocs that have joined this year, and they seem to be enjoying it. The idea is that it's an expanded version of what we're going to be doing over the next few weeks. It's a bit more advanced. We cover most topics in more detail, and we cover more topics. This is a mixture of lectures and practicums, and the idea is you can come away from it knowing how to do advanced sequencing analysis of both short and long read data sets in a multitude of contexts. Hopefully, if you've taken this course that we're doing now, that will get you the prerequisites you need, including working in high-performance computing environments and having experience working in R-based environments. The other course I run in conjunction with Kelly Ruggles is called Advanced Integrative Omics. This is a really fun course. We deliberately have a very small class size, and the idea is there's a single project to which all members are contributing. Most of the work gets undertaken in small groups, so you typically work with one or two other people, and we keep switching the groups around every few weeks. It's a student-led course in the sense that we don't have formal lectures. All we do is we present a problem and a whole bunch of data sets, and then you will take them away and try to wrangle them into something that can be something useful that we can then write a paper about and hopefully get published. The prerequisites for this are, of course, more advanced. You need to have a lot more experience working in HPC environments and R-based environments, and you also need to have a demonstrated prior experience working with different sequencing data types. But if either of these are of interest to you and there's something you want to do as part of a forthcoming master's or as part of your grad school program, then feel free to drop me emails if you want to know more or talk to David. But setting the scene for today, what we really want to talk about is how high throughput sequencing has fundamentally changed how we approach science. It's become a kind of a very common readout for many different types of lab experiments. Most papers you pick up today will have at least some kind of sequencing in it. It seems to be almost a prerequisite in cases. Clinical and basic science investigators from all aspects of biology are now using this technology regularly. The problem, of course, is that many people still remain very naive about bioinformatics, and particularly the analysis of sequencing data and the complexities that come with that. We're also faced with the good thing, which is that sequencing costs are coming down dramatically, which now means that there's increased use in routine assays. There's a lot more technical innovation and a lot more novel applications that are going on in this field. All of this raises additional challenges. The most of which is that sequencing informatics has very much become a bottleneck. The sequencing itself is easy to do. You can pay companies, you can take your samples to the Genome Technology Center at NYU, give them money, basically give anyone money, and they will run off and sequence things for you. The problem is that they will then give you the raw data back, and it's up to you to figure out what you have to do with it. And this is really the key message here, is that sequencing informatics is the essential part of this, the essential point of this science. It's really about data analysis and also trying to discover meaning in the results that you generate. One of the big advances in sequencing technology is that more and more samples can be sequenced at ever greater depth. But what that means is the amount of time and the cost of analysis is also going up. This is a slide I often like to show to people, which is my view of what the beauty and challenge of sequencing informatics is. And it really comes from the expression that there are many different ways to skin, or in this case, shave a cat. There is no single right way of analyzing sequencing data. There are many wrong ways, and I'm sure, like me, many of you will become experienced with the many wrong ways over the course of the next few weeks, and indeed years if you stay in this field. But always be assured that just because your solution seems to be different to other people, it doesn't necessarily mean it's wrong. It just means you need to think about it a bit more carefully, and don't assume that because someone else has a nice solution that looks nice and shiny, that it's necessarily correct, because often it is not. One of the big challenges of working in the field of sequencing informatics is very much staying in the game. Hopefully you'll recognize this picture from Alice in Wonderland, running to stand still. This is a problem in the sequencing world simply because there is a rapid turnover in technology platforms. I've actually lost count of how many different versions of Illumina sequencing platforms there have been. There have been other technologies like 454 and Ion Torrent that have come and to some extent gone. Each of the different vendors, each of the different major technology companies have different standards for what they think data should look like or how accurate data should be. There's also a rapid evolution, particularly these days with something called nanopore sequencing, which I'll talk about later. There is this rapid evolution in how sequencing approaches are undertaken, and also in what you can do with the data. The other challenge you will find from more of an informatics point of view is there is so much software out there, most of which is only used a few times and then dropped. Others of which are very popular in the wider community, but are often updated. And a lot of them tend not to be very well supported because they're open source distributions and they don't have dedicated people who can spend their time fixing every little bug or updating the software to work with every new little bit of technology that's released. So this means there's a lot of kind of fighting against the flow, as it were, in terms of trying to keep your data analysis up to date and trying to keep your pipelines functional. The other problem is, as sequencing becomes cheaper, as I said before, you get much more data, much more bang for your buck. But the problem is that the more data you have, the longer it takes to align it, the longer it takes to process it, and everything can start to slow down. And this just is a challenge you have to accept is always going to be there, and you're never going to find a perfect solution to it. So moving into today's topic, I wanted to start off with just a very brief history I'll talk about the history and the future of nucleic acid sequencing. And really it's a one-slide story of, well, a couple of slide stories. So how did we get from the 1970s and Fred Sanger, who was shown on the left, who invented Sanger sequencing, to where we are now with technology that can be taken to space, it can be taken to the Antarctic, can be taken to the top of the mountains and the bottom of the sea, and which is smaller than most of your cell phones. And this is, of course, the Oxford Nanopore sequencer, which you can see on the right. Excuse me. So for today, we're going to go through this abridged history of sequencing. I'm going to talk a bit about why we sequence. And then I'm going to introduce you to the three major technologies. I've got some little videos to show you as well, just to mix things up. So we'll be talking about Illumina short read sequencing, PacBio long read sequencing, and Nanopore long read sequencing. And then at the end, I'm going to throw out a few cautionary words on experimental design, which hopefully will save you a lot of money one day. So this is my sort of favorite figure regarding what the history of sequencing looks like. You can trace it all the way back to the mid-1800s when Gregor Mendel figured out the fundamental principles of heredity, and then we can move up through the different years. I've highlighted in orange what I think are the kind of the major events that are worth considering. So in 1975, Fred Sanger introducing the plus and minus method of sequencing, and then very shortly after dideoxy sequencing, which is the basis of Sanger sequencing. And then that really was the workhorse of the molecular biology world for the longest time. But then once you hit the early 2000s, you got the launch of 454, which is a high throughput sequencing, pyrosequencing platform. And you also had the launch of Illumina, who as a company had just bought out another company called Solexa, who invented what is now known as the Illumina sequencing method. These have been very successful. 454 started to fade away after a few years, although it still had a lot of interesting uses. But Illumina, I'm sure most of you have heard of, and it's really become the workhorse of the field. More recently, a couple of companies, PacBio Sciences and Oxford Nanopore, they launched very new technologies aimed at performing what's called long read sequencing. We'll talk about the difference in short and long read sequencing later on. But really, the main message to take from this is it's really been the last sort of 10 to 15 years that the sequencing world has kind of leapt forward, and that we now have this incredible wealth of technology with which we can ask probably more questions than we can imagine even at present. And that is a hugely exciting place to be. So going back to 1975, we had Fred Sanger, as I say. He got 2 Nobel Prizes for his work, principally on the idea of DNA sequencing. On the right, you can see an example of his DNA sequencing using the plus-minus method. This was when they were run out on gels, and you can imagine having to take a gel like this, having to look and identify every band and write it down as being an A, a G, a C, and a T. Now, it's bad enough if you have to sequence something small like phi X, which is a few thousand characters, but you can imagine as you go to bigger and bigger fragments of DNA or bigger and bigger genomes that this rapidly became impossible. Nonetheless, it was still a hugely exciting idea. And what this really led to was the idea of dideoxy chain termination, or the Sanger sequencing method. And the simple idea behind it is that, let me just get those right. The simple idea behind it is that you can generate DNA fragments of different lengths. You can terminate these with labeled nucleotides. And depending on where the termination occurs, depends on where you'll see a fluorescent band. So basically when you have your DNA fragment, you'll add in typical nucleotides such as DATP, DCTP, DTP, and TTP, a DNA polymerase, your DNA fragment of interest, and your primer will basically bind to a region of the DNA that you're interested in, and then these bases will begin to become incorporated by the DNA polymerase, creating copies. Now, if you add small amounts of a dideoxynucleotide to these reactions, then it basically means every time a fragment is copied, it will randomly stop when a dideoxynucleotide is incorporated. So that basically means you produce a huge number of fragments that will all start at the same position, but will terminate at different positions along the fragment that you're interested in. What you can then do is look at this using fluorescence and basically identify, depending on the fluorescence, depending on the antibody that's used, whether it's an A, whether it's a G, whether it's a C, whether it's a T. And you can also then look at this on a gel or in a sort of capillary electrophoresis style apparatus to work out what the order of the bases are. And that is kind of shown in this schematic. So the question is, or at least one question I get asked is why we sequence. And the reason why we sequence is because it can tell us a huge amount about the relationship of DNA and RNA and protein within cells, within systems, in context of virus-host interactions, in context of cancer, you name it. We know now that DNA encodes RNA and RNA encodes protein, but we also know that there is a huge amount of regulation that occurs in this, that RNA can interact with RNA, that there are many different RNA species, including messenger RNAs, microRNAs, non-coding RNAs, circular RNAs, and so on, and that these can serve many different functions instead of being made in, or as well, or instead as being made into a protein. RNA can directly regulate protein activity, it can directly regulate DNA activity, it can also regulate other RNAs, it can also regulate itself. So sequencing gives us the ability to actually probe all of these sorts of interactions and to better understand. So you could design an experiment where you have an RNA of interest and you could say, well, I want to know which proteins this RNA binds to, or which other RNAs this binds to, or where in the DNA genome this RNA is binding to. You can design an experiment, you can generate fragments, you can sequence them, and you can infer information from that. So with the development of 454 sequencing, and particularly Illumina sequencing, we had the birth of high throughput sequencing. And the idea is, rather than sequencing individual fragments by PCR one at a time, that we can now potentially sequence whole organisms or whole transcriptomes all at the same time. It gave us the power to do de novo genome sequencing, so basically to go out, take any plant, any creature, anything that you want, to take its DNA, to break it into small pieces and sequence it and reassemble it. It gave us the ability to do things like exome sequencing, which is often used in cancer studies, looking at copy number variation. It gave people the ability to go into the tombs of old Romanian monasteries and find corpses that were mummified many hundreds of years ago and sequence them to see what the human genome looked like at that time, or to sequence bacteria or other pathogens that are still present. There were some interesting studies where people were looking at tuberculosis and what the genome of tuberculosis looked like 500 years ago. You can also move into, of course, RNA-seq and whole transcriptome analyses. So that allows us to generate a much better understanding of gene structure and a number of different RNA transcripts that may be encoded, the way that alternative splicing works, the way that microRNAs are produced. And then, of course, there was epigenomics, which is a very big one. So understanding how chromatin control is regulated, how methylation is involved in these things, basically how transcription is controlled at the chromatin level. And then most recently, of course, metagenomics has become a very big thing. So the idea of whether it's environmental metagenomics to kind of see, do we have plague on the New York subway, or do we have COVID-19 now on the New York subway, or whether it's just going into individual people and looking at the way that diet may impact your internal microbiome, or what happens to your microbiome if you go spend a few days on the International Space Station. So all of these are questions that people can ask and that they can now relatively easily answer thanks to the advent of high throughput sequencing. So I'm going to start off with some examples, just a very brief introduction to short read sequencing and then a video. The principle of short read sequencing is quite simple. You will start with either DNA or RNA that you're interested in. It could be a fragment that you've PCR amplified. It could be RNA you've extracted from an organism. It could be the genome you've extracted from an organism. But once you've got your genomic DNA or you've captured your RNA, you need to make cDNA from the RNA, and then you basically need to do fragmentation. Now, typical fragmentation is done using acoustic sonication, which is great because it, in theory, induces random shearing. By tuning the parameters of the sonicator, you can generate fragment sizes of whatever you desire. The optimal range for short read sequencing is about 150 to 300 nucleotides. There are some other strategies out there, which include the use of transposases or targeted ligation, but we won't go into those in any particular detail today. Once you've fragmented your DNA or cDNA of interest, you then need to do some basic end repair to prevent degradation, and then you will ligate adapters onto the end of it. And these adapters are absolutely crucial for the sequencing reaction, as you will see from a video I'll show you shortly. Once you've done this, you will typically, not always, but typically you will do PCR amplification, which will allow you to enrich for fragments that are correctly ligated. And this is done basically by having PCR primers embedded within, PCR primer targets embedded within the adapter sequences that you ligate. Once you've done this, you've generated what we term a sequencing library, and this is ready to then go on a sequencer and be sequenced. So let me just show you a short video, and this will give you what I think is
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
Okay, so hopefully that gives you at least a brief overview of how this works. I think a couple of people have already asked whether the lecture material will be available. I think we hope to get this up on the course website afterwards, and I will add in some links so people can find these videos again if they're interested. Now, I think one of the main messages, or the main message I would want you to take away from this is the bridge PCR step. Obviously, the folding step is extremely important in the Illumina sequencing process, but it's also highly restrictive. Simply put, they haven't been able to make that fold happen with fragments that are longer than about 700 nucleotides. And that means that no matter what you do, Illumina will always be a short read sequencing approach, at least for as long as it requires this bridge PCR step. So anytime that you want to generate sequence data using fragments that are longer than that, you're going to have to find a different platform. But we'll move on to that more in a minute. The point I want to make with this slide is that if you go on the Illumina website, and again, I'll add the link to this slide later on, you can download several different documents which look like the one I've shown on the right-hand side of the page. Each individual line you see in that document is detailing a different type of seq strategy that is used for DNA. And this can be everything from regular DNA-seq to mRNA-seq to gDNA-seq to about 100 or more other ones that are on there. And really the main thing to take from this is that while the sequencing strategy is exactly the same, the number of different ways you can prepare your samples in advance, particularly the way you can try to capture samples or capture perhaps interaction points between different types of RNA and DNA, or between different types of RNA and so on. There is a huge range of things that you can do there. So I definitely encourage you to visit the Illumina website and look at some of these. Anytime you're developing your own platform or your own method, it seems like you can give it whatever name you want as long as it's followed by seq. If you're lucky, maybe one day it will also be incorporated into documents like these. But one of the things you need to remember is every time you use a slightly different type of sequencing preparation technique, it's a different way of building libraries. So it's probably going to have some impact on your informatics analysis downstream. The way perhaps that you're going to QC the data, the way you're going to align it, maybe the software that you want to use to do the analysis. So you just always need to bear this in mind, that every time you use a new technique to generate libraries, there's probably going to be something different required in the analysis downstream. My computer's stopped. There we go. Another thing I just wanted to bring to your attention is the fact that Illumina has a range of different sequencing platforms. I've shown what the popular series are that they have here, and underneath I've written what the data output from a single run is. Now you can see that you have little benchtop systems like the Mini-seq, the MySeq series. They produce comparatively small amounts of data, particularly relative to the cost of running them. But in some cases, they are very useful. You can imagine if you're working with small organisms, or if you don't have many samples, quite frankly, you don't need that much data. And it would become very efficient to put on some of the high-scale sequences like the HiSeq or even the NovaSeq. The NovaSeq particularly is really designed for sequencing human genomes or for multiplexing many different samples. But this is also, as was hinted at in the video, part of the power of Illumina sequencing is this idea of pooling samples. So I mentioned before that you add adapters onto the end of the sequence fragments that you generate. And as you saw from the video, these adapters are then used in the sequencing itself. Now, within these, there are several different strategies you can use to add these adapters and also to add indexes within them. So one is you can have a standard adapter that you add to every single fragment. And then when you do your PCR amplification, you make sure that your primers for a given library have a unique piece of sequences in them, which is referred to as a barcode. What this means is that you can generate lots and lots of different libraries separately, each of which has a different barcode. And then you can pull them together and sequence them all in a single run. And then you can use various types of software to kind of deconvolute the data afterwards and separate them out. And this is an incredibly nice and incredibly clean way of putting lots of samples together and reducing the cost. And there's no doubt that in terms of cost and efficiency and data output, that Illumina remains by far the best in this area. Okay, so then I want to go on to long read sequencing now and share a few more words with you on that. If I can get my thing working. So long read sequencing is something that's really come about in the last probably 3 to 4 years that has really taken off. It was initially very, so one of the challenges of long read sequencing is that throughput tends to be a lot lower than short read sequencing. But it was very much embraced by the microbial community because it was a fantastic way of sequencing viruses, bacteria, and other types of microbes. If you're willing to put enough time and a significant amount of money into it, you could also use it to sequence and perform de novo assembly of eukaryotic genomes. The principal advantage to using long-read sequencing is that where you have what are termed problematic genome sequences, such as repeat regions or other low complexity regions, these are virtually impossible to resolve with Illumina sequencing because the read lengths are very short. But with long-read data, where the read lengths are long, often over 1,000 nucleotides, sometimes much longer, these sorts of regions can be completely resolved. There's also a very effective technology if you want to look at things like structural variants, or if you want to look at rearrangements, which is something that's very difficult to capture with short read sequencing. And this then extends to the mapping of long insertions or deletions, detection and mapping of mobile elements that you may find. And perhaps most excitingly, at least from my point of view, the idea of doing direct detection of methylated bases, or indeed any base that carries some kind of modification. And then also, particularly an interest of mine, working in the field of transcriptomics, is the idea of rather than taking RNAs, breaking them into small pieces, sequencing them and trying to put them back together, is now you can actually sequence transcripts in their entire length, all the way from the 5' cap to the poly A tail. And this provides kind of an unparalleled resolution in terms of looking at transcript isoforms, understanding patterns of splicing and alternative splicing and fusion transcripts and many, many more things. Perhaps the simplest visual of how this would work is on the top of this graphic. I'm showing you what short read data would look like. You can see you're generating many, many more reads, but they are significantly shorter, and trying to find overlaps between them is going to be harder. When you generate long read data, you get far fewer reads, but they are significantly longer, and actually finding overlaps and joining them together becomes, in theory, a simpler problem. With this table here, I've tried to highlight what I think the major advantages of each technology is. So with short read sequencing, you get a higher read depth, and it used to be much, much higher, although now, at least with Nanopore, they have some platforms which are which are generating reads at numbers that are getting close to being comparable with Illumina. But for now, Illumina is very much king. Read lengths in Illumina are very short. I mentioned the constraint on fragment sizes, but typically read lengths are going to be somewhere between 50 to 150 nucleotides, whereas with long read platforms like PacBio and Nanopore, you can go into the thousands, if not tens of thousands of nucleotides. And in fact, Nanopore doesn't have an obvious upper ceiling. And there are a group of people out there who spend a lot of time trying to see if they can outdo each other in terms of generating long reads. And at least the last time I checked their website, the longest Nanopore read that had been generated to date there's around about 2.2 megabases, which is frankly astounding. In terms of looking at RNA, transcript resolution is much higher with long read platforms than with short read, principally because each individual read has the potential to be representative of an RNA in there, rather than having to computationally reconstruct them as you would with short read data. Similarly, de novo assembly is generally a lot easier with long read data, although in truth, most people employ hybrid strategies where they overlay long read data with short read data. The relative cost, short read data is definitely going to win on that for now, although it's starting to get better in terms of long read sequencing. Recoding bias. So with short read data, you always need to recode RNA into cDNA for your analysis. But with Nanopore in particular, they now have protocols that allow you to natively sequence RNA from a sample, which is of course incredibly powerful. In terms of input requirements, short read sequencing technologies generally don't need very much. You can start with pico or nanograms of total RNA, whereas with longer read approaches, you often need micrograms. Again, some of that is changing depending on the protocols you use, but it does mean that long read approaches aren't always super great to use in kind of clinical settings without having kind of prime manipulation of your template by, for instance, PCR or such. The versatility of Illumina sequencing is incredibly high, as I hopefully made a point of before when I talked about all these different library preparation techniques that are out there. It's also incredibly high for Nanopore and PacBio sequencing, and I'm sure in another year or two, the orange block highlighting short read sequencing data is very much going to move across the long read data as people do more and more exciting things with particularly the Nanopore technologies. So we talked a bit about the workhorse in terms of Illumina sequencing. This is one that everyone does. I want to show you a quick video about how PacBio sequencing works. PacBio sequencing has very much got its place. I'm not sure as a technology how much longer it's going to last in comparison to Nanopore, but there are certainly some places that still very much swear by it.
Introducing the PacBio Sequel II system, powered by single molecule, real-time, or smart sequencing technology. Here's how smart sequencing works. First, from any sample type, ranging from viruses to vertebrates, DNA or RNA is isolated. Next, a SMRTbell library is created by ligating adapters to double-stranded DNA, creating a circular template. Primer and polymerase are added to the library that is placed on the instrument for sequencing. At the core of smart sequencing is the SMRT cell, which contains millions of tiny wells called zero-mode waveguides, or ZMWs. A single molecule of DNA is immobilized in the ZMWs, and as the polymerase incorporates labeled nucleotides, light is emitted. With this approach, nucleotide incorporation is measured in real time. With the Sequel II system, you can optimize your results with two sequencing modes. Use circular consensus sequencing mode to produce highly accurate long reads, known as hi-fi reads. Or use the continuous long read sequencing mode to generate the longest possible reads.
Okay, so hopefully that gives you a brief idea of how PacBio sequencing works. It's certainly very different to Illumina. It's a really, really clever idea. The biggest problem with it is that it's just not very high throughput. I mean, most PacBio runs that you do are going to generate somewhere between 500,000 to 1 million reads, and that's something which doesn't seem to be scaling up particularly well. Initially, it had a lot of problems with error rate, and this is in fact something that plagues long read sequencing technologies, although this is something they're able to significantly reduce with their circular consensus sequencing. It's also an exciting technology in terms of its applications. It was used a lot for transcriptomics early on. It was very powerful in that case. But there's also been interesting, or at least some interesting studies, particularly this one, which I'm highlighting here, where people have used it to look at sort of complex rearrangements and amplifications of oncogenes in breast cancer cell lines. And this is a particularly good paper to read if you're interested in learning more about kind of PacBio sequencing and its potential role alongside short read sequencing approaches in tackling these sorts of questions. But that's pretty much all I'm going to say on PacBio sequencing, principally because I don't work with it very much, and also because I think it's been superseded by the final technology that I'm going to introduce, which is nanopore sequencing. I've got one last video for you to enjoy. Okay, so that gives you a brief overview of nanopore sequencing, and if you don't know what you just saw, I will try to explain it to you in a little bit more detail now. So the principle of nanopore is very much that you generate these protein nanopores, which at the moment are taken from different types of microbes, although they are working on generating some synthetic versions. And you basically embed these in an electrically resistant polymer membrane. And that's kind of shown, where's my mouse? That's kind of shown over here. You then embed you then within this membrane have an array of microscaffolds, where each of the scaffolds supports a membrane and its embedded nanopore. And then you also have a sensor chip associated with each of these microscaffolds, which means that you have individual control over every single nanopore that you have embedded within the wider membrane. And what this means is that you can monitor what each nanopore is doing individually at each time. And this is controlled by something called an ASIC, and we don't need to go into that into great detail. But principally what it means is, principally how it works is you can take a piece of DNA or RNA, in this case, hopefully not fragmented. In the case of, say, RNA, you could ligate a specific adapter to the poly A tail, or if it's DNA, you could do some very gentle shearing to produce fragments that are tens, if not hundreds of kilobases long, and that add adapters onto the ends of those. Those adapters are then tethered to what's called a motor protein. And when you put it on the device itself, the motor protein is attracted to these protein pores. They will attach, and the motor protein is in fact a helicase. So it will unwind your double-stranded DNA, or it will unwind your RNA, which is generated with a cDNA hybrid. And that RNA or DNA will then be passed through the nanopore. During this time, you also have a current flowing through the nanopore. And depending on the bases, the exact composition of bases that are present within a certain region of the pore at a certain time, the current will change. And you can basically read this as is shown in the schematic on the left, as depending on where the current is, you can say it's likely to be an A, or it's likely to be a G, C, T, and so on. And that's going to show in live action on the right through the GIF. Now, as you can imagine, this is a very powerful technology. It's particular value in my book is the fact that you can sequence native DNA or RNA. And if you do, and there are modifications that exist within the DNA or RNA, such as 6MA, M6A, or 5MC, those modifications will also have a slight impact on the current. So for instance, you could distinguish between an adenosine that is methylated at the N6 position versus an adenosine that is not. So potentially, and probably still a couple of ways from being able to do this perfectly, but you would be able to take a piece of DNA or RNA, sequence it, and not only see what the bases are, but also determine which modifications are present. And the ability to do all that in a single sequencing reaction is absolutely incredible, because it's something that if you did it with another technology, you would need to do a number of different library preps and sequencing reactions, a lot of different analyses, whereas Nanopore offers you the potential to do everything all in one go. The way they do this is through a number of different devices that they've generated. Probably the most commonly known one, the one that you'll have hopefully seen before in lectures or presentations or so on, is a Nanopore MinION. I have one of these myself plugged into my computer at work. It fits comfortably in the palm of your hand. It's about half the size of a cell phone. On top of that, you have smaller devices such as a flongor, which fits into a MinION by way of an adapter. They've also been working on something called a smidge ion, which, as you can see, is something that would plug into the bottom of the mobile phone. And the idea is that can be used in diagnostic settings or out in the field if you're doing environmental sampling or so on. And then they also have things called a grid ion, and then more recently the PromethION, which are really designed to be the kind of the high throughput platforms that they hope will rival Illumina one day. And certainly with the latest incarnation of the Promethean, you can generate, tens if not hundreds of millions of reads from samples. So it becomes a very powerful system. Then they also have other interesting features such as Voltrax, which is this automated library preparation system, which is particularly useful again if you're working in the field somewhere or in a non-sterile setting. They have what's called the MinION Mark 1C, which is basically a computer with a MinION device built in. So you don't need to rely on having another computer to plug things into. And then they also have something called a Minute, which is basically a very small version of a sequencing laptop, which you could also plug your device into. So all of these have become very popular and they get used in all sorts of different ways. Now, the MinION as standard is probably going to be used on benchtops and labs all over the place, or in diagnostic clinics, but it has been taken to a number of different places. So in the top left, I'm showing you a picture generated by folks who work in the Antarctica, in the dry valleys, and really what they're doing down there is doing environmental sampling, but they're also trying to see, can you get sequencing platforms working live on location in places where you have sub-zero degrees, sub-zero Celsius degrees. The bottom left, I'm showing you a classic photo from what was known as the Zebra Project, and this was during the Zika outbreak in Brazil. A whole bunch of scientists got together, rented a couple of buses and started driving around the Brazilian countryside, sampling people wherever they could find cases and just running and sequencing, tracking the outbreak live. And something similar was done during the Ebola outbreak as well a few years back. And it proves a very useful way of kind of tracking outbreaks while they're in progress. If people are interested in learning more in terms of the current COVID-19 outbreak, there is a website called oh, this is embarrassing, I've blanked on it. It's called Arctic Network. It sounds like Arctic, but it's not. It's Arctic Network, ARTIC. And this is basically a UK consortium that have put together protocols for sequencing directly from patient samples on the Nanopore, and then capturing the virus and immediately uploading it to NextStrain so they can again track outbreaks almost in real time in terms of seeing where infections are coming from, where they're going, and how they're spreading in the community. Perhaps most famously, if you look in the upper right-hand corner, Nanopore is the only sequencer that has been to space and worked successfully. This is the astronaut Kate Rubins, who took a Nanopore up there on, I think it was her last mission. And she was taking DNA from mice and viruses and bacteria and showing that you could sequence those in zero gravity conditions. This is, of course, extremely exciting when you consider potential missions to the moon or to Mars. The ability to, track the health of astronauts during travel and after moon landings and so on, but also potentially to do environmental sampling. And then the last picture is to show the MinION on my desk, where I try to work with it as much as possible. Okay, so that's giving you a brief overview of the major sequencing technologies. For most of the rest of this, we're going to focus just on short read sequencing, so we're really going to be just focused on the Mumina. But if you do want to learn more about long read sequencing, then I do encourage you to check out the courses I mentioned at the start. I'd like to finish up with a few thoughts on experimental design. So this is just a classic example I use. Consider a situation in which you want to identify M6A modified bases in RNA. This is something many people seem to be doing these days. There are a number of different methodologies that exist. So the first thing you need to decide is, well, which of these methodologies do I want to use, which is most appropriate to my sample? Do I want to go down the Mumina pathway, which is probably very well designed, very well supported. Lots of the informatics has been worked out already, but perhaps it's not as sensitive, or perhaps it's not as precise as if you were to go down a PacBio or a Nanopore route. But if you go down the latter two routes, then maybe your informatics needs to be more customized and you need to be able to deal with more complex data sets. So my general advice to everyone, which hopefully you always think about whenever you're doing any kind of experiment, is, what is the actual biological question or questions that you're trying to answer? If it comes into sequencing, do you need biological replicates? Do you need technical replicates? And if so, how are you preparing these and how are you controlling for things like batch effects? When you're doing sequencing, one of the first things you need to think about is, well, how many sequence reads do I need to get the answer I want? Can you guesstimate? Can you more precisely calculate? Depending on that, you need to think about, is my protocol amenable to multiplexing? Is this something where I could sequence lots of samples together to bring down costs and potentially to generate more usable data sets, or is it something I'm going to have to do kind of one at a time? And this then also links to things like, what resolution do you want? Do you just need to know the region within, say, 20, 30 nucleotides where a modification may be, or do you want to know the precise base? How important is linkage information? If you have two modifications that are 1,000 nucleotides apart, you wouldn't be able to tell whether they exist on the same piece of RNA with the Mumina methodology because it's short read. But potentially with something like the Nanopore or PacBio system, you might be able to capture that. And then, of course, probably the most important one is budget. So you always need to plan for that. And then even before you start with that, you need to think about software for analysis. What kind of software is out there? Are you going to have to write something yourself and test it and then try to get it published and deal with reviewers who don't like it? Are you going to have to, is it something you get working in your local computer? Does your local computer have the capacity to handle large sequence data? Or do you need it working on a HPC environment like Big Purple? And if that's the case, then is your software compatible with it? Do you have test data sets you can use to make sure your software is working? Do you know which parameters need tweaking? Because you will always need to tweak parameters. So these are just a few things I think you should keep in mind. I've put a link at the bottom that gives you some more thoughts on that. You also need to bear in mind confounding factors such as batch effects, and batch effects, which are basically the plague of my life and most people's lives. These can occur during nucleic acid extraction, and also during library preparation. So you need to be quite careful about how you plan to do those and how you can try to minimize the batches, and also how you can record potential batch effects as they're happening so that you can informatically try to model them and to correct for them. Finally, I have a list of rules that I think people should live by when it comes to working, designing sequencing projects. This is more aimed at informaticians, but actually I think it probably applies to everyone. You always want to make sure you're part of an experimental design meeting, ideally during the early stages of the project. Whether you're an informatician or just a sequencer who's going to be using someone to do the informatics, you need to make sure there is good communication. You need to make sure everyone knows what they're trying to do, what data they're trying to generate, what it's going to look like, and then what can be done with it. There's nothing worse than blowing 20K on a sequencing experiment and then having an informatician tell you, well, you can't answer the question you want with this data, you should have done it this way. And I've been in those situations and people are very upset when you tell them you've wasted money. You also need to bear in mind there's a lot of existing data sets out there, more and more every day, of course. A lot of these can be used to answer questions without having to go away and actually do your own sequencing. It's often used a lot if you're trying to generate prelim data for grant applications and so on. But you always need to bear in mind that if you're using other people's data, you need to spend a lot of time trying to understand how they generated it. I've tended to find method sections of papers can be a bit dry in terms of details. So often you end up having to chase people to try and find out more, but you should never be afraid to do that. Where possible, always try to make sure nucleic acid extractions are carried out using the same kit and batch, ideally at the same time or at least closely together. And you also need to think, if it can't be all done together, you need to find ways to group your samples so that batch effects can be minimized. Randomization before library preparation is always a very good one to do. I actually do that myself. I trick myself and I create a document and I randomize things, and then I don't look at it until after I've made my libraries to try and make sure that I don't introduce biases myself. You also need to bear in mind that, whether or not you're doing library preparations by hand, or if you have someone else doing them, or you have a machine doing them, you need to ask for some kind of tracking database that keeps an eye on where samples are relative to each other. And this can really help deal with problems like aerosol contamination, which unfortunately can be a problem in some situations. You'll hear more about demultiplexing at a later date, but basically always be very strict in your demultiplexing to make sure you don't mix samples from different batches. And in general, just always be careful when you're dealing with low frequency observations. Whether using short or long read data, you will often find that, if you see a mutation or if you see an RNA expressed at low levels, a low frequency mutation, a lot of the time this can come from errors in the sequencing or from the assembly or alignment of data. So it's always worth spending a bit of extra time kind of looking at this more closely, and if possible, trying to eyeball the data yourself to convince yourself and others that what you're seeing is really real. So that just about brings us up to the end of our time together. I'll be speaking to you again on Thursday when we're going to go over sequence read data formats, principally FASTA, FASTQ, and FAST5 type data formats. I'm going to talk to you a bit about single and paired end read data structures and what this means. And then I'm going to be introducing something called the sequence alignment format. This might all sound a little bit dry, but it's absolutely a crucial step in kind of learning about sequencing informatics. If you don't know what the difference between these files are, if you don't know how to parse them, if you don't know what all the different things that you can find in the SAM file mean, you're going to have a lot of problems in your downstream analysis. So that's it. Thanks for your attention. I think there may be some questions in the chat box. If not, then I'm sure you can ask some, or if you want, you can put your hand up and we will try to get to you and answer some questions.